Now guys, moving on to our next point. We know guys, enzymes are proteins. So they are actually a big structure. But in this big structure, there is something, a cleft. And this cleft we can call as substrate binding site or the catalytic site. Fine, so now guys, remember one thing, that active site of the enzyme, which may be called as substrate binding site or the catalytic site, is actually complementary to not the substrate shape, but the shape of transition state. Right? Remember this line. Now, let's talk about the basic properties of active site. Now, because active site has specific amino acids present in them. For example, we have a group of enzymes called as serine proteases, like trypsin, elastase. These are a group of serine proteases. Some blood coagulation factors are also serine proteases. So they have a specific triad of serine, histidine, and aspartate at the active site, which participates in the chemical reaction, which participates in the catalytic mechanisms of these enzymes. So one thing, that active site has specific amino acids which are catalytic, right, which are helping in the catalysis or speeding up the reaction. Like serine proteases have serine, aspartate, and histidine triad. Right, guys? And one thing I told you, that transition state is more complementary to the active site than the shape of substrate. Okay, so shape of substrate is not important. The shape of transition state is more complementary here. Remember this. Now, how does the substrate binds to the enzyme? How does it bind? Is there any covalent bond? No. Guys, most of the time covalent bond is rarest possibility. So what type of interactions are involved? Hydrophobic. Yeah, non-covalent interaction. What you can use the term? That when the substrate is binding to the active site of enzyme, it is binding through the non-covalent interactions. So what are the non-covalent interactions? Like electrostatic, van der Waals forces, hydrogen bond, hydrophobic interactions, etc. So these are involved in the active site. Now is active site, and we have a term called as allosteric site. They are different. Allosteric site, suppose this is the enzyme. So allosteric site may have present here. It may be present somewhere else. So that is not the part of active site at all. Active site may also binding to the cofactors. Guys, cofactors, we will just in few minutes we will talk about that. But cofactors are helping the enzymes for their catalysis, right? Like metals or the vitamins, etc. Fine. Now, moving on to the next point, guys, most of the time during the reaction or during the catalysis by the enzymes, what is happening? It is happening in the cell. Now, cell has an aqueous environment. Water is there, right? So water may sometime interrupt the reaction. Let's say, for example, hexokinase. What does hexokinase do? As we all know, glucose to glucose 6-phosphate. Who is the donor of phosphate? ATP. Where is the phosphate added? Like to the OH group of the glucose. Fine. So water also has a OH group. Fine. So if the hexokinase is working and ATP is the source of phosphate, water is present at the active site, then what will happen? This may waste the ATP because phosphate may be transferred to the water, OH group of water instead of glucose. So that is why, guys, most of the time when the reactions are happening, when the catalysis is happening, water is removed from the active site. And this term is called as desolvation of active site. It's a very important term that you should know. Desolvation of active site. Correct? Fine. Now let's move to our next point. Guys, active site may have specificity for particular, substrate, especially in terms of shape, let's say, right? So that can be like the shape specificity. They can have a specificity based on the stereospecificity or the chirality. For example, we have amino acids oxidase, L amino acid oxidase, D amino acid oxidase. So that is a specificity of the enzymes which is based on chirality or the stereospecificity, right? Then guys, some enzymes may have the specificity or active site have specificity according to the bonds it has to break. Like for example, amylase only breaks alpha-1,4 glycosidic bond. It doesn't act on the alpha-1,6 glycosidic bond or other bonds. Fine. So such kind of specificity may be present at the active site, right?